Today we're going to talk about a molecule that is absolutely fundamental to your health and the process of aging. This molecule is called NAD, which stands for nicotinamide adenine dinucleotide. While that name might sound complex, its role is something we can all understand because it is central to how every single cell in your body produces energy. Think of NAD as a critical currency for cellular operations. It is not the energy itself, like glucose or fats, but rather the essential facilitator that allows your cells to convert that fuel into usable power. Without sufficient levels of NAD, the entire system of energy production would grind to a halt, impacting everything from your muscle movements to your brain function. NAD exists in two primary forms within your body, NAD+ and NADH. You can conceptualize these two forms as a tiny, rechargeable battery that operates within each of your cells. NAD+ is the uncharged or oxidized form, ready to accept electrons during metabolic processes. When it accepts these electrons, it becomes NADH, the charged or reduced form. The constant cycling between NAD+ and NADH is what drives the creation of ATP, or adenosine triphosphate, which is the main energy molecule that powers nearly every activity in your body. It acts as a helper molecule, or coenzyme, for a vast number of enzymes. Imagine a factory full of highly specialized machinery. NAD+ is like the universal power adapter that allows many of those different machines to plug in and run. One of its most critical roles is acting as a coenzyme for a class of proteins called sirtuins. You may have heard of sirtuins before, often referred to as longevity genes. These proteins are powerful regulators of cellular health, and they are entirely dependent on NAD+ to function. Think of sirtuins as the master regulators or the maintenance crew for your cells. They are involved in processes like DNA repair, inflammation control, and maintaining the stability of your genome. Another essential family of enzymes that rely on NAD+ are the PARPs, which stands for poly-ADP ribose polymerases. PARPs are the first responders to DNA damage. When a strand of your DNA breaks, PARPs rush to the site of the damage and use NAD+ as a substrate to signal and recruit other repair proteins. This is an incredibly energy-intensive process, and it consumes a significant amount of the cell's NAD+ pool. Every time a PARP enzyme fixes DNA, it uses up a molecule of NAD+. This creates a direct trade-off. The more DNA damage your cells experience, the more NAD+ is diverted to repair, leaving less available for energy metabolism and sirtuin activation. One of the most consistent and well-documented findings in the biology of aging is that NAD+ levels systematically decline as we get older. This isn't a small drop. Studies in both animals and humans suggest that by middle age, your NAD+ levels may be half of what they were in your youth. This decline is a multifactorial process, meaning it is driven by several interconnected factors that compound over time. Understanding these drivers is key to understanding why aging is so closely linked to a decline in cellular function and an increased vulnerability to disease. One major reason for this decline is an increase in the activity of an enzyme called CD38. You can think of CD38 as the primary consumer or eater of NAD+ in the body. While it has important roles in the immune system, its activity tends to increase significantly with age. This creates a vicious cycle. As NAD+ levels drop, the activity of sirtuins, which also help protect DNA, decreases. This can lead to even more genomic instability, triggering more PARP activity and further depleting NAD+, accelerating the downward spiral. While aging is the most universal cause of NAD+ decline, it is crucial to understand that certain disease states and lifestyle choices can significantly accelerate this depletion, regardless of chronological age. Many chronic metabolic diseases are characterized by impaired energy metabolism, and at the core of this dysfunction, we often find compromised NAD+ levels. Chronic inflammation is another powerful driver of NAD+ depletion. As we touched on earlier, inflammation activates immune cells, and these activated immune cells ramp up their expression of the NAD-consuming enzyme CD38. This creates a feedback loop where low NAD+ impairs sirtuin function, further promoting inflammation, which in turn consumes more NAD+. Lifestyle factors also play a profound role in modulating your NAD+ levels. For example, a diet high in processed foods, sugar, and unhealthy fats can induce metabolic stress and inflammation, thereby increasing the rate of NAD+ consumption. Excessive alcohol consumption is particularly taxing on the liver's NAD+ pool. On the other hand, certain lifestyle interventions are known to naturally support or increase NAD+ levels. Caloric restriction or fasting has been shown in numerous studies to be a potent activator of NAD+ synthesis and sirtuin activity. Similarly, regular physical exercise, both endurance and resistance training, is a powerful stimulus for increasing NAD+ in muscle and other tissues. Given the critical role of NAD+ and its decline with age, a major focus of scientific research has been on finding ways to safely and effectively boost its levels. This has led to intense interest in NAD+ precursors, smaller molecules that the body can use as raw materials to synthesize NAD+. One of the most well-studied of these precursors is nicotinamide riboside, or NR. NR is a form of vitamin B3 that is naturally found in trace amounts in milk and other foods. It serves as a direct building block for NAD+ through what is known as the salvage pathway, which is the body's primary mechanism for recycling and creating new NAD+. The appeal of NR lies in its efficiency. Alongside NR, another precursor has gained significant attention in the scientific community and the public sphere: nicotinamide mononucleotide, or NMN. NMN is also a derivative of vitamin B3 and is the immediate precursor to NAD+ in the salvage pathway. This has led to a scientific debate about which precursor is superior for supplementation. The central question surrounding NMN supplementation has been how it gets into cells. For a long time it was thought that NMN was too large to cross the cell membrane directly. However, more recent research has identified a specific transporter protein in some tissues that appears capable of moving NMN directly into the cell. The vast majority of our current understanding of the benefits of boosting NAD+ comes from an extensive body of research conducted in animal models, primarily in mice and rats. These preclinical studies have been instrumental in establishing the proof of concept that raising NAD+ levels can combat age-related functional decline. The findings from these studies are often striking. For instance, in numerous experiments, supplementing aged mice with either NR or NMN has been shown to significantly improve their physical performance. Mice given these precursors can run longer and farther on treadmills, demonstrating enhanced muscle stamina and mitochondrial function. Beyond physical endurance, animal studies have painted a compelling picture of systemic metabolic benefits. Supplementation with NAD+ precursors has been shown to improve insulin sensitivity and glucose tolerance in mice on a high-fat diet, suggesting a potential role in managing metabolic syndrome. Furthermore, studies have documented improvements in cardiovascular health. The neuroprotective effects of NAD+ boosters in animal models are also a major area of excitement and ongoing research. In models of neurodegenerative diseases like Alzheimer's, raising NAD+ levels has been shown to reduce the buildup of toxic protein aggregates, improve cognitive function, and enhance synaptic plasticity. The crucial next step after the promising animal research was to test NAD+ precursors in humans. Over the past several years, a growing number of human clinical trials have been conducted to assess the safety, bioavailability, and efficacy of supplements like NR and NMN. The first and most fundamental question these trials had to answer was, does oral supplementation with these precursors actually increase NAD+ levels in the human body? The answer, from multiple studies, is a clear yes. Controlled trials have consistently shown that daily supplementation with commercially available doses of NR and NMN can significantly increase the concentration of NAD+ in the blood and other tissues like muscle. With bioavailability confirmed, the focus has shifted to whether this increase in NAD+ translates into tangible health benefits for humans. The results here have been more modest and nuanced compared to the dramatic effects seen in mice. Some studies have shown positive, though not transformative, outcomes. However, many other human trials have found no significant effect on key outcomes. For instance, several well-conducted studies looking at physical performance, insulin sensitivity, and body composition in older or overweight adults have found no difference between the group receiving an NAD+ precursor and the group receiving a placebo. Whenever we consider taking a new supplement that has potent biological effects, it is absolutely critical to have a thorough discussion about safety and potential side effects. For NAD+ precursors like NR and NMN, the safety profile so far appears to be quite favorable, especially in the short to medium term. Human clinical trials have generally found these supplements to be well tolerated. However, it is crucial to emphasize that the majority of these studies have been relatively short in duration. The long-term safety of chronically elevating NAD+ levels over many years is still an open question. One theoretical concern relates to the role of NAD+ in cell growth, leading to a hypothesis that boosting NAD+ could potentially fuel the growth of pre-existing undiagnosed cancer cells. The current state of human research can be summarized as promising but incomplete. We know that these supplements can successfully increase NAD+ levels. We have some initial signals of modest benefits in areas like cardiovascular health and inflammation. However, the evidence is not yet strong enough to make definitive claims about reversing aging, extending lifespan, or treating specific diseases in humans. The future of NAD+ research is incredibly bright. We are still in the early innings of understanding how to best leverage NAD+ biology for human health. For now, NAD+ stands as a central pillar in the science of longevity, a testament to how understanding fundamental biology can open up new pathways for enhancing our health span and improving our quality of life as we age.